Hi guys, today we're talking about the evolution of phototrophs or photosynthesizing organisms. So like we talked about on the previous video over the earliest life on Earth, this first life might have been a simple chemotroph or a simple phototroph. Phototrophs get their energy from light and chemotrophs get their energy from chemicals. And in that video, we assumed that that first organism would have been a chemotroph and then talked about the different types of organisms that could be. But in this video, we're going to be talking about the evolution of phototrophy, which may have predated chemotrophic evolution, but in this video, as well as the previous one, we're going to still assume that chemotrophs were first, and now talk about the evolution of phototrophs after the chemotrophs and how they might have expanded from potentially hydrothermal vent origin environments. But first, I must point out that the first phototrophs were likely not these sunflowers or any sort of plant. Actually, they were microbes. I talk more in my Life Origins video about endosymbiosis and how plant cells came from endosymbiosis, or basically merging of two cell types that eventually worked as one organism, and before plants was microbial phototrophs. So what was the first kind of phototroph? Was it the around 3.5 billion year old cyanobacteria that likely made the stromatolite? Or was it whatever caused this 3.9 billion year old formation to have light carbon isotope signatures? If you want to know how light carbon isotope signatures indicate life, you can watch my carbon isotope video. I'll link it up here for you if you want to check it out. But actually, the first phototroph was probably neither of these. Well, it might have been whatever caused the light carbon isotope signatures in this rock here, but there's a distinction I need to make between oxygenic phototrophs and anoxygenic phototrophs here. So oxygenic phototrophs are those that you're probably most familiar with, things like plants, things even like cyanobacteria that take up carbon dioxide and use it and light energy to split water and make oxygen. And then they release the oxygen, and then animals like ourselves breathe in oxygen and release carbon dioxide. It's a cycle. However, these types of phototrophs that produce oxygen were likely not first. Anoxygenic phototrophs, those that produce compounds that aren't oxygen, for example, instead of producing oxygen from water or O2 from H2O, there are anoxygenic phototrophs that take H2S or hydrogen sulfide and produce sulfate or SO4. So they don't actually produce O2 or oxygen. That's why they're called anoxygenic. And these likely evolved first, but I'll show you why that is later in the video. First, let's just back up a second and talk a little bit about why phototrophy might have been beneficial for early organisms. So assuming a hydrothermal vent origin of life, chemotrophs would have started first at that dark hydrothermal vent and used chemicals to gain energy, but then eventually life would have wanted to venture out into new environments, but it was restrained by needing the chemicals that were coming out of the hydrothermal vent. So how did they eventually move away from the hydrothermal vent? Well, one possible way is using a different energy source. Instead of the chemicals coming out of the vent, using light, for example. One primitive type of pigment in microbial cells was likely bacteriorhodopsin, which basically captures light energy to transport protons out of the cell. This created a proton gradient, more protons out of the cell than in the cell, creating a proton pump, which basically pumps protons through a protein in the membrane of the cell called ATP synthase, which basically pumps the protons through the protein, creating ATP as it does so. And ATP is chemical energy for all of life. Our cells in our body create ATP through a proton pump, and we use that ATP to do our daily things. And they could have used this rhodopsin to make this proton gradient to create ATP. However, early on, it might have been present in chemotrophs as an alternative or extra source of energy for when light was present, but maybe the chemicals they wanted weren't super abundant. And so in low nutrient environments and conditions, they were like, well, we need some energy, so let's use this rhodopsin as backup to to absorb light energy and make ATP that way, since we don't have the chemicals we like right now. But all other phototrophs use chlorophyll or bacteriochlorophyll as their light-absorbing pigment rather than rhodopsin. So it's possible that phototrophy evolved twice, once with rhodopsin and once with chlorophyll, but it's more likely, or I've seen more references that say that the iron core of rhodopsin proteins was eventually replaced by magnesium, allowing more efficient light-sensitive pigments called bacteriochlorophylls. We can see in the figure down here that chlorophyll, which isn't the same as bacteriochlorophyll, but I'll explain that later, absorbs different wavelengths of energy, both lower and higher, than bacterial rhodopsin does. And these more efficient absorbing pigments would have increased microsurvival on early Earth because they became thermotactic. This means that they could detect hydrothermal sources, and that's because many of these bacteriochlorophyll pigments were capable of absorbing infrared radiation, which anything that gives off heat gives off IR radiation, so that's how they could detect these sources. However, you might be thinking, earlier I mentioned that if they were able to use light for energy, why do they need to be near hydrothermal sources still? Well, even though they could use light for energy, they still needed an electron donor, like molecular hydrogen or hydrogen sulfide and other things, and all of these electron donor compounds came out of hydrothermal vents. So they had to still be near the hydrothermal vent fluid to get their electron donors that they used the light to convert to something else to gain energy. However, later I will discuss when certain phototrophs evolved, they no longer needed to be near hydrothermal vents because their electron donors that they needed were not coming from the hydrothermal vents, but more on that later. Eventually, an early version of what's called the reaction center complex used in phototrophs evolved from the adaptation of mobile ubiquinones. Ubiquinones are basically compounds in all living cells that act as electron-carrying coenzymes for the electron transport chain. If you've ever taken a Bio 101 class, you've probably heard the term electron transport chain. It's exactly what it sounds like. It's a chain of reactions and electron-carrying compounds that basically move electrons down this chain of compounds until it creates something that can help make ATP for the cell. The end goal is always ATP. And in phototrophs, this reaction center complex allowed electron flow from the excited pigment. So the pigment like chlorophyll, for example, gets excited by light, and electrons from this excited pigment were carried by electron-carrying compounds and enzymes to kick off the electron transport chain. But what type of organism was all of this occurring in? What was the first phototroph? We already said it wasn't a eukaryote, so we could cross that off the list, but was it part of bacteria or archaea? Well, the lack of photosynthetic archaea basically crosses off archaea from that list, and we know from that that photosynthesis likely evolved from the bacterial domain. However, that's pretty much the only thing that's agreed upon when it comes to what the first phototrophs actually were. Some genetic evidence suggests that green non-sulfur bacteria were the first phototrophs. However, some suggest heliobacteria and other evidence suggests purple bacteria. So which one was first? Well, if we look at this figure, we can see some bubbles listing off these bacteria, and all of these little abbreviations with numbers next to it are certain genes that some of those bacteria had and where they might have come from. And it's trying to make a flow chart of which phototrophic genes belong to what and where did they come from. And we can see heliobacterium here, that's the heliobacteria. We've got the purple bacteria over here, and chloroflexus is a type of green non-sulfur bacteria. So they're all listed here, but the flow chart isn't very unidirectional. It's kind of all over the place, and the reason for this is lateral gene transfer. You guys know I talked about lateral gene transfer, or LGT, in my previous video over the earliest organisms. Same thing happened with chemotrophs, and it just makes everything a big mess when we look at genes from primitive organisms and try to map phylogeny. It's never a straightforward map of who came first and whatnot. It's always very confusing because lateral genes transfer, or the transfer of genes across species boundaries, makes it hard to know if those genes were being passed down or just transferred laterally across species. So we may never know exactly which of these groups of bacteria are responsible for the beginning of photosynthesis, and making lateral gene transfer even more detrimental was microbial mats. Basically, microbial mats are layered mat-like structures, like I showed earlier with the stromatolite, that basically has microbial communities literally right next to each other, stacked on top and underneath one another, because they benefit from each other's metabolites and they end up stratifying themselves in this, layered structure. I talk about microbial mats in a whole video. I'll link it up here if you want to check it out. But that facilitated lateral gene transfer because one, close proximity to other communities of microbes, and also EPS. So EPS is like an extracellular mucousy type substance that microbes excrete for a number of reasons and functions, one of which is the transfer of genetic material, which EPS facilitates. So that also made lateral gene transfer a real pain in the butt for phylogenetic purposes. So we don't know, but one of these three groups is potentially responsible for the first phototrophs. So let's talk about them. First, purple bacteria, basically bacteriochlorophyll, that pigment we talked about earlier, is used in purple bacteria and absorbs near infrared radiation. And purple bacteria can be broken up into both purple non-sulfur and purple sulfur bacteria, named basically because one uses sulfur and one doesn't. And so purple non-sulfur bacteria can use hydrogen-2 or molecular hydrogen as an electron donor and CO2 as an electron acceptor, and make basically organic carbon, this is glucose, and water as their products of their metabolism. And the way that they kind of set off this reaction is by the light absorption. So they use light as energy to do their metabolic pathway. And purple sulfur bacteria basically do something similar, but instead of molecular hydrogen, they use hydrogen sulfide. And because purple non-sulfur bacteria do prefer using molecular hydrogen, this may indicate that they originated near hydrothermal vents where molecular hydrogen was abundant. So if these were the first photosynthesizing bacteria, then they likely got their start either at a hydrothermal vent or some sort of hydrothermal system where molecular hydrogen would have been available. After the first phototrophs, or purple bacteria, or potentially heliobacteria, or potentially green non-sulfur bacteria, then green sulfur bacteria might have come next. I put acteria here. Pretend that has a B and it says bacteria. My bad. These guys might have come next, and we know that they were a tad more advanced because they had a new reaction center complex that contained iron sulfur clusters and were more electronegative upon illumination. And so more electronegative means the light excited them more, allowing more electrons to be transported through the chain, aka more energy created. These also could grow in what some phototrophs and other organisms likely considered toxic environments, because green sulfur bacteria liked sulfur and not only could grow but liked to grow in very sulfidic and anoxic, without oxygen, environments like the lower layers of microbial mats. And I talk more about that and the kind of stratification and who is where in microbial mats in my microbial mats video that I mentioned earlier. So if you want to know more about that, you can see that video. Eventually, though, all of these bacteria, green non-sulfur bacteria, purple bacteria, heliobacteria, green sulfur bacteria, all of these phototrophs would have grown together, likely grown together in microbial mats, which is precisely why there was rampant lateral gene transfer. But moving on, these mats were potentially still near hydrothermal sources because, again, they needed those electron donors for their metabolism, but they also had to be shallow enough for light to penetrate. Now, remember that the photozone, how deep light penetrates in the ocean, goes to around 200 meters in depth in the modern ocean, and likely was similar in the early ocean, maybe even more, I don't know, but it goes pretty deep. So you could have had a hydrothermal area that also received light pretty easily. So this is where they likely would have been. Again, they had to use those compounds that came out of the vent fluid, like hydrogen sulfide, molecular hydrogen, iron sulfur, etc. And I promise you later we will get into how they finally broke away fully from the hydrothermal areas. It did not need that vent fluid anymore. But before we talk about that, let me briefly go over photoprotection. Basically, because O2, or molecular oxygen, was not abundant enough on early Earth to make ozone. I talk more about when Earth was oxygenated in my Great Oxidation Event video and my Neoproterozoic Oxidation Event video, but basically there weren't oxygenic phototrophs to make oxygen with their photosynthesis photosynthesis yet. And so because that's the major source of oxygen on Earth, oxygen hadn't really become that abundant yet. So because it wasn't abundant enough to make ozone, UV light could get through the atmosphere instead of being stopped by ozone like it is today. And it reached Earth's surface and also penetrated into the ocean. And it could have been harmful to those early phototrophs, and they would have had to evolve protection mechanisms because, as we see here, UV light is harmful to DNA. And everybody on Earth has DNA. So, like, we need to not have it, damage our DNA. So, mats might have been not only beneficial because they got the metabolites from their, sister community, and they were like, oh, thanks for the food. And also thanks for some genes too. I like that as well. But they also would have been beneficial because it protected at least the lower layer communities below the surface layer from UV light. But they also could have used sediment as a sunscreen rather than their bacterial friends. Ferric minerals or oxidized iron minerals would have been perfect sunscreen for this UV light because they absorb light in UV light wavelengths around 220 to 270 nanometers. And amorphous silica could have also been used as a sunscreen for UV light because they would have both absorbed UV wavelengths while simultaneously allowing essential wavelengths that the phototrophs wanted and needed through to get to the phototrophs and so they could use it. And we actually have plenty of examples in modern organisms as well as ancient ones of the secretion of amorphous silica for shells and skeletons and whatnot. And because this secretion of silica is so common, yet sometimes a little enigmatic because it doesn't make sense to try and use silica for shells or skeletons when calcium carbonate is so much easier to secrete because of its saturation state in the ocean. However, this, protection, this sunscreen type mechanism or function might have been one reason that evolved. And last, for photoprotection, I just want to mention carotenoids. So carotenoids are just great for photoprotection. They are not like the primary pigment for absorption of light in phototrophs, just like I talked about, the primary pigment is bacteriochlorophyll or chlorophyll. And so carotenoids are accessory pigments in all phototrophs that are important for just basically protecting from excess or harmful light. They also protect against the harmful products of photosynthesis, like oxygen radicals. And so they kind of act as antioxidants. And then not only are these carotenoids great for photoprotection, but also they attract pollinators and seed dispersers in plants, and they also assist in absorbing light in low light conditions. Now finally getting to the switch from bacteriochlorophyll to chlorophyll. Bacteriochlorophyll and chlorophyll are both pigments used in phototrophs, but they absorb different wavelengths of light. Bacteriochlorophyll can absorb longer wavelengths of light, and chlorophyll absorbs anywhere from around 430 to around 680 nanometer wavelengths of light. And shorter wavelengths of light are actually higher in energy. And the higher the energy, the more stable compounds the organism will be able to break down through their metabolism that is charged by light energy. So with all this energy, chlorophyll-using organisms were able to oxidize and split these super stable molecules of H2O or water. So cyanobacteria were these new kids on the block that were able to use chlorophyll to split water. Why was this such an incredible feat? This splitting of water produced oxygen, molecular oxygen, and this was the beginning of oxygenic photosynthesis. And not only was this monumental because oxygen was produced and eventually that led to us being able to evolve and live because we breathe oxygen, but this was also monumental because since water is unlimited, instead of being limited to hydrothermal vent fluid, it was literally everywhere in the ocean. It's made of water. This allowed cyanobacteria to expand from their hydrothermal origins or potential hydrothermal origins and diversify throughout the entire protoxone of the ocean, including continental shelves and shallow water environments near the ocean. And it was just an amazing expansion of life because it wasn't limited to this source of hydrothermal vent fluid or the chemicals that came from it. Cyanobacteria evolved around 3.5 to 2.7 billion years ago. 2.7 is the most undeniable evidence for cyanobacteria. 3.5 is more of a probably these microbial mats represent cyanobacteria, but we're not 100% sure. But in either case, those two numbers are both way before what we call the Great Oxidation Event. I mentioned this earlier. It's basically when Earth became largely oxygenated because of oxygenic photosynthesis, which was done by cyanobacteria. So why didn't the oxidation event happen right after or like during the evolution of cyanobacteria? Because cyanobacteria evolved and immediately expanded because of their ability to use water rather than chemicals from hydrothermal vents for their metabolism. Well, the reason why the GOE or Great Oxidation Event didn't happen until way later, or around 2.4 billion years ago, is probably because of chemical reservoirs. I talk more about this in my GOE video, but basically these cyanobacteria produced O2, right? Molecular oxygen, but then it's releasing this oxygen into a really reduced Earth. And I mean chemically reduced. That just means that there are a lot of compounds out there that could have been oxidized by this oxygen, aka they could have reacted with it to transform into something else. And because of this need for oxygen to go and react with and oxidize all these reduced compounds out there, it had to fill these reservoirs of chemically reduced species before it could build up on its own. I don't know why I'm talking like this, but I am. It's just that this is so cool to me. Anyway, yes, I'm a nerd, I know, but basically you had to fill these chemical reservoirs to be able to eventually then build up in the atmosphere and oceans. So this buildup here basically marks the point where, boom, all those chemical reservoirs are basically filled to the point that oxygen could then build up on its own without reacting with a bunch of things. It was like, oh, yeah, I've already reacted with you. I can just be my own thing now. And although molecular oxygen might seem precious to us and all animals on modern Earth today, on early Earth during the Archean, it was a toxic byproduct. I mean, it was just the waste of these photosynthesizers, and their expansion was so quick and widespread, and they created this huge oxidation event that it actually caused a lot of extinctions, because many of these early anoxygenic phototrophs and anaerobic chemotrophs not only didn't like or use oxygen, but they also, many of them couldn't tolerate it. It was toxic to them. So a lot of them died and went extinct, or at least were extremely restricted to the corners of the earth and ocean that weren't oxygenated at that point. But eventually, organisms did evolve to use molecular oxygen in their metabolism, obviously. So we will talk more about the many aerobic respiration metabolic pathways, which there are more than one, more than just the way we do it. There are plenty of aerobic respiration pathways that use oxygen in metabolism. And we'll talk about those in the next or an upcoming GeoBio video. I'm not sure if it'll be the next one. Anyway, if you want to check out the references I'm using to make this video and many other videos in my GeoBio playlist, you can check out Geomicrobiology by Kurt Konhauser. This is a great book. I'm using this for a lot of the videos in this playlist, as well as the Brock Biology of Microorganisms. So those are two great references. They're listed and linked in my description below. And lastly, in the upcoming videos, other than the aerobic respiration evolution video, we'll talk about extremophiles and all the variations of extremophiles, those organisms that love extreme environments, or at least what counts as extreme to us humans. And then we'll also talk about when life moved to land. I know I did a poll on my channel and you guys really liked that idea, so I will talk about that in a future video as well. And until then, you can check out the playlist below, the geobiology playlist, which is shown up, or at least will show up on the screen here in a second. So check that out if you want to see any other geobiology videos, or see if these videos are out by the time you're watching this. And I will see you guys next time. Thanks for watching. Bye.